Sodium dodecyl sulfate, polyacrylamide gel electrophoresis, SDS Page, is a method used for the separation of large biological molecules. A variation of this technique, photo-induced cross-linking of unmodified proteins, Picup for short, allows for the detection of multi-protein assemblies known as oligomers within a sample. Here's a demonstration of the cross-linking process in action. The cross-linking reaction involves the introduction of a photosensitive agent as well as an oxidizer to the protein. Once the sample solution is mixed with both chemicals, it is exposed to light for a set period of time as achieved with the SLR camera body. The photosensitive agent is raised to an excited state when stimulated by the photons. It then donates an electron to the oxidizer. The now positively charged photosensitive agent strips an electron off of a particular amino acid in the protein. The protein stabilizes itself by sharing an electron with a neighboring protein, forming a covalent bond. This process cross-links oligomers. An additional chemical is introduced to stop any further reactions. The samples are now ready to be prepared for gel electrophoresis. The proteins must be able to travel through a mesh-like gel, so in order to do that, the samples include SDS, a detergent used to break apart any aggregates or clumps of proteins. They are also heated to unravel or denature the proteins. After a few minutes at near boiling temperature, the samples are centrifuged to condense any evaporated buffer. Note that pickup is necessary to maintain oligomers, or else the detergent would simply break them all up. An electric field is applied across the gel. In order to conduct current, the apparatus must be submerged. The proteins are now ready to be loaded into the gel. Gels are generally close to 1 millimeter in thickness and several centimeters in height and length, with notches at the top into which to pipette samples. Each sample is placed in one of these notches, referred to as lanes. Here you can see small bubbles forming due to the ionization of the liquid touching the current-carrying wire. The proteins will navigate through the gel at a velocity dependent on their charge and molecular mass. As a consequence, different proteins, or in our case oligomers, will travel different distances, separating the initial sample out into discrete bands. Once the gel is finished running, it is ready for development. The hard casing must be removed and the gel must undergo a series of wash cycles to produce an image visible to the eye. The process is much like that of developing a photograph in a darkroom. First, a fixative is added to hold the proteins in place, preventing any further movement throughout the gel. A synthesizer is then added to allow the proteins to be stained. Once stained, the gel is then developed. The bands will begin to appear as seen here. When the desired exposure is achieved, stopper is added to finalize the process. The gel is placed between two transparencies to be able to be scanned. The gel is scanned into a computer and is ready for analysis. The ladder on the side is a reference tool of proteins of known molecular mass and charges to help identify what is present in the sample. The darkness of the bands is representative of their relative proportion.